planet is full of harsh and extreme environments, from the freezing Arctic to hot arid deserts to high altitudes where the oxygen levels are low and the exposure to UV is high. Yet despite these harsh environments, life persists, whether it's us as humans, animals, plants, microorganisms, and with all these different species adapting to the same harsh environments, it begs the question, can we find shared adaptations across species? Can we potentially find the same adaptation in people who live in Tibet as we do in their domesticated chickens, as we do in the yeast in their wine? And of course, yeast do not have lungs like we do, but what we do all share is that fundamental unit of the cell. And that brings us to something we call cell autonomous adaptations, or CAAs. So often when we think about adaptations in a multicellular organism, we'll often focus on adaptations in a tissue or organ, perhaps a change in our heart or our lung. And in fact, a famous example in Tibetans at high altitudes is altering their levels of erythropoiesis, or red blood cell production. And this process involves our kidney, bone marrow producing cells, something that's coordinated and clearly not cell autonomous. But on the other hand, researchers found that in the Sherpa people in the Himalayas, the mitochondria within their muscle cells are more efficient at utilizing each and every oxygen molecule when they compare them to the muscles of non-adapted lowlanders. So what we see here is two adaptations to the same stress that are occurring at different biological levels. And we are interested in these cell autonomous ones, and a fair question to ask is why? So one big advantage of focusing on CAAs is that they're experimentally accessible. So if you want to study a coordinated organ tissue level adaptation, it often requires moving to mouse models, which can be long and laborious experiments, and then there are species differences. But with CAAs, we can test them directly in human cell culture in the lab. It's faster, more scalable. And this is not only important when you want to investigate an adaptation, but particularly if there's some kind of therapeutic potential. Second, from an evolutionary standpoint, it opens new questions. If I'm an organism adapting to a new environment, do I first adapt with cellular changes that then give me time to coordinate a more organ-level adaptation? Or perhaps it's the opposite. And can understanding this teach us something about multicellular evolution? And finally, as I'll aim to show you in this talk, just by making this distinction and reanalyzing data, we're able to find novel candidates for adaptation to high altitudes that may have been otherwise overlooked. So we focus in on high altitude in this project because it's one of the most extreme environments that humans live in. And we ask, can we detect CAAs in human populations living at high altitude? And can this approach reveal novel, overlooked adaptive traits? So to do this, we combine two complementary methods. And the first one intuitively starts at the cellular level. So we take human skin cells, we expose them to UV radiation, and we ask which genes help the cells survive this stress. Now, I know what you probably are thinking, that high altitudes, the famous stress is low oxygen levels, but another major stress they're experiencing is UV radiation. And this is a map of our globe that is colored by UV exposure, pink being the highest. And I've marked some well-known high-altitude environments, particularly the Papua New Guinean highlands and the Andean Plateau are exposed to some of the highest UV exposure on our planet. So the idea behind our experiment is this. We have cells, and we delete, let's say, gene X, and we can either expose these cells to UV radiation or no UV. And we can track their growth over time. So if we don't see a difference between the growth, whether we expose them to UV or not, or maybe there's no real change over time, just fluctuations, this is the pattern we expect of a gene that is not essential for UV radiation. However, if we delete gene Y and expose it to UV radiation, and we see that the frequency drops in the population, they're suffering from this deletion. But when we delete the gene but don't expose them to UV, they do fine. That's the pattern of a UV essential gene. Now, these are just theoretical gene examples, but we set up an experiment that enables us to do a large-scale experiment and ask this question about all 20,000 human genes. So to give you an example of what real data looks like from this experiment, this is the gene ERCC8. So this is a gene that's central in the nucleotide excision repair, which is the major repair pathway activated under UV stress. And we see when we knock out this gene, I'll say that this diagram is similar to what we saw on the previous slide. The x-axis is progression of time, or generations, and the y-axis is the frequency. So when we knock out this gene and expose it to UV, as shown in the orange lines, the cells are dropping in frequency by orders of magnitude. They are suffering. But in the gray, when we don't expose them to UV, they do great. So this is one of our UV essential genes. Now, of course, we cannot look at all 20,000 human genes in this way, so we had to develop a quantitative framework that enabled us to ask this about all 20,000. And ultimately, we were able to find 154 genes that are essential for the cells to survive this UV stress. Now, great. We found what genes are important for the cell, but that doesn't yet tell us whether they're important for the organism. Were these genes ever used as a solution in evolution to adapt to high altitudes? And for that, we turn to populations that have been living at high altitudes for thousands of years in an environment that most of us would struggle in. We work with published studies that identified adaptive signals in their DNA by comparing the DNA of these high-altitude populations to the DNA of non-adapted low-altitude populations. And our goal is to take these adaptive signals, link them to the genes that they're affecting, and ultimately, we want to overlap both of our approaches and find genes that we see are both important for the cells to survive, as well as linked to adaptive signals in high-altitude populations. Now, a challenge we first have to face is actually linking these adaptive signals to the genes that they're affecting. So only 2% of our genome is actually encoding genes and proteins. 98% of it is what's called non-coding DNA. Now, this DNA includes a lot of important information like when and where genes express, but it's still more difficult to interpret. And so this causes a key question. With so many adaptive signals falling in this non-coding DNA, we need to ask, what genes are they affecting? So to tackle this, we worked with a great tool developed by the Golomb lab by Yarden and Simon that essentially integrates multiple large-scale data sets to be able to enable us to ask that question. And so we used this tool on 3 geographically and genetically distinct high-altitude populations: Tibetans, Andeans, and Papua New Guinea Highlanders. And essentially, we generated a list of genes that are linked to the top adaptive signals in these populations. Now, these populations live in different parts of the world. They've been largely adapting independently for thousands of years to this stress, but they are experiencing similar stresses. So we first wanted to ask whether we could find shared adaptive signals amongst them. So when we looked for overlaps between the adaptive signals in these 3 populations, we found almost nothing. But once we linked those adaptive signals to the genes they are predicted to affect, then we saw highly significant overlaps across the populations. So what that suggests is, while the genetic changes might not be shared across these populations, some of the genes that they are affecting are shared. In fact, there are 13 genes that are linked to top adaptive signals in all 3 populations. Cool for us to see that 2 out of them are important for UV-induced damage response. Okay, so back to our question. We found the genes that are important for cells to survive UV. We found the genes that are linked to top adaptive signals in high-altitude populations. And when we overlap them, we found 20 candidate adaptive genes. Now, to get an idea of who these genes are, we did some functional grouping. We see things like DNA repair, stress response, signal transduction, transcription regulation. These are core cellular processes. Now, I'm going to show you 2 gene examples. The first one is ERCC8. You may remember that is the gene that I showed you earlier on as our UV essential gene from our screen. But it is also linked to top adaptive signals in Papua New Guinea Highlanders, which, as I said earlier, are exposed to some of the highest UV on our planet. Now, this gene to our knowledge, has not yet been suggested to be adaptive in humans at high altitudes, but it has come up in studies looking at Tibetan boars, as well as plants at high altitudes, which I think beautifully demonstrates this idea of cell autonomous adaptation, that plants that have a totally different organ system than us could share that same adaptation as us. And the second gene is RPS2. This is a ribosomal gene. Ribosomes are what produce our proteins, one of the most fundamental processes, so it was surprising to us. It doesn't seem like an obvious UV candidate. However, recent research has found that ribosomes play an important role in UV response. So UV is not only damaging our DNA, but it's also damaging our RNA, and the ribosomes are some of the first to encounter this and notify the cell to respond. So here, what this suggests is that adaptations to high altitudes can also include fundamental cellular processes. So I want to leave you with the following key takeaways. The first is that cell autonomous adaptations are a powerful but still underexplored mode of adaptation. We performed a genome-wide screen that identified 154 genes as essential for cells to survive UV. And by combining approaches, we revealed novel adaptive candidates that have both functional support and evolutionary support. And finally, by better understanding the evolutionary solutions to extreme environments, this could potentially inspire new therapies for human disease. So I want to finish by thanking my advisors, Stachy Pilpel, Esseke Willerslev, Orna Dahan, and everyone who helped shape this project. I want to thank the organizers for this wonderful interdisciplinary event, and thank you for listening.